All right, so we're going to kind of tackle those three situations: the glycolysis, the taking care of triacylglycerols, and digesting proteins and amino acids kind of different. We're going to start with glycolysis. This is all about glucose, 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 glucose. All right. You can see there's this nice little flow chart here. I took this directly from the book. Like this is not, I didn't make this. All right. And so here we have is it's a linear 10-step process. So we like start with A and we get to the end. All right. It revolves around glucose. Glucose is converted to two molecules of pyruvate. There's an anaerobic pathway. Steps 1 through 5 that's where we like invest energy to create things. And then steps 6 through 10, that's where we get energy out. And so we're going to break those down like real specific. All right. You can see the next slide is 1 through 3. So glucose is our principal monosaccharide. Here's our beta anomer, and then this is our straight chain. It's not a ring yet. All of this goes down in the cytoplasm. All right. So steps 1 through 5, we're adding 2-phosphate. The addition of 2 phosphate groups requires energy from ATP, so we get energy there. The cleavage of a carbon-carbon single bond makes 2 3-carbon products. And so that's what we're getting here. We're taking glucose as 6 carbons, we're chopping it in half so we have 2 3-carbon things. Step 6 through 10, this is where those 3-carbon products are oxidized. We form NADH, 2 phosphate bonds broken, and ATP is formed again. NAD plus converts those CH bonds to CO bonds, and then NAD plus is reduced to NADH, and phosphorylation of ADP to ATP needs energy. There's a whole lot going on, so we're going to have to break this down like stepwise. All right, so we're going to start with steps 1 through 3. And the reason that we're doing this stepwise is because now we can look at these and say, okay, let's look at each structure and what's happening at each time. So here we start glycolysis steps 1 through 3. We're gonna start with glucose, no big deal. In this first step right here, we have phosphorylation of glucose. ATP becomes ADP, so we dump a phosphate group onto glucose. All right, you can see that here we just have, we got our phosphate group out here. All right, see there's our phosphate group. See, this is where I get that P with a circle. It's just very simple. All right. Step 2, so here to here, we get what's called isomerization. All right. Isomerization from the 6 ring to the 5 ring. So glucose 6-phosphate becomes fructose 6-phosphate. All right. That's the idea there. All right. So we change this to this guy right there. All right. So glucose isomerizes to fructose. Then we have a second phosphorylation. Here we go. 3. Boom. Right there. ATP becomes ADP. So we have another addition of phosphate. This requires energy, and we get fructose 1,6 biphosphate. So we go from here to here. Right. So here, we phosphorylation occurs, then isomerization, and then another phosphorylation. In the next video, I'm going to jump into the next steps.